Hello guys, today we will talk about antibodies and antigen. This education activity is dedicated to the spirit of my mother. Here is a definition of antibodies. Antibodies are a circulating protein that are produced in vertebrate in response to exposure to protein structure known as antigen. An antibody is a mediator of humoral immunity against all classes of microbes. The substances that generated or were recognized by antibodies were called antigen. There are three types of molecules that used by adaptive immune system in order to react to antigen. One of them is antibody, secondly the major histocompatibility complex, and finally the T-cell antigen receptor. Antibodies are synthesized only by B-cell lymphocyte lineage and exist in two forms: the membrane-bound antibodies and the secreted antibodies. The membrane-bound antibodies function is working as B-cell lymphocyte activation, as it mentioned here, and the secreted antibodies function as neutralizing agent for toxin and prevent the entry and spread of pathogen and eliminate microbes. Here is general function of antibodies at all: neutralization of microbe and toxin, activation of the complement system, ozonization or cooking of pathogen for enhanced phagocytosis, and activation of antibody-dependent cell-mediated cytotoxicity, which is used by innate immune system, and antibody-mediated mast cell activation to expel parasitic worms. That in this 5 type is antibody-mediated mast cell is belonging to type 1 hypersensitivity. Here is some definition: antiserum, any serum sample that contains detectable antibody or non-antibody molecule that bind to particular antigen. Here, if you have a serum and the serum contain non-antibody against non-antigen, that serum is called antiserum. The serology, the study of antibody that and their reaction with antigen. Here we want to say a note that healthy 17 kg adult human produce about 2 to 3 grams of antibodies every day. Almost 2/3 of this is an antibody called IgA, which is produced by activated B cell and plasma cell in the wall of gastrointestinal respiratory tract. And because IgA is produced in this gastrointestinal respiratory organ, it have a huge amount, because gastrointestinal respiratory tract have a large surface area. Here is the 3 molecules that used by adaptive immune system in order to make an immune response: the immunoglobulin and T-cell receptor MHC molecule. According to antigen binding site, the immunoglobulin bind to antigen by variable heavy and variable light domain, and the T-cell receptor bind to antigen by variable alpha and variable beta domain, and the MHC molecule bind to antigen by alpha 1 and alpha 2 domain in class 1 MHC, and alpha 1 and beta 1 domain in class 2 MHC. Nature of antigen is a macromolecular protein, like protein and lipids, polysaccharide, and small chemicals, as well as small chemicals. The T-cell receptor antigen is peptide which is binded to MHC complex, and the MHC complex antigen is peptides. Nature of antigenic determinants is the linear and conformational determinant. Here we can see that the determinant is the 2 type of determinant, the linear and conformational. Here in T-cell, the linear determinant peptide, about 2 to 3 amino acid residues, which is binded to MHC molecule. In MHC molecule, the linear determinant of peptide only some amino acid residues of a peptide. The affinity of antigen binding is most powerful in immunoglobulin because it have less dissociation constant concentration in order to make an constant interaction with antigen. And the loose one is a T cell receptor. It have more concentration in order to make a constant interaction with antigen. The antibody structure is quite different according to the cells that produce it, as in multiple myeloma, a monoclonal tumor of antibody producing plasma cell often have a large amount of biochemically identical antibody molecule. General feature of antibody structure is contain some feature. One of them is plasma or serum protein can be physically separated based on solubility characteristics into albumin and globulins. Gamma globulin is found in the third fastest migrating group of globulins. Another common name for antibody is immunoglobulins, referring to the immunity conferring portion of the gamma globulin fraction. The immunoglobulin term and the antibody term is used interchangeably in this book. All antibody molecules share the basic same structure characteristic but display a remarkable variability in the region that bind the antigen. Here is the region that bind antigen. The antibody molecule has a symmetric core structure. Here is a symmetric core structure composed of two identical light chain. Here is the light chain in green, and two identical heavy chain. Here is the two identical heavy chain. Here is the heavy chain and here is another. Both the light chain and heavy chain contain a series of repeating homologous units. Here is the homologous unit called the immunoglobulin domain. Here is the immunoglobulin domain. It consists of 110 amino acids. An immunoglobulin domain contains two layers of beta sheet. Here is the only one domain. It contains two layers of beta pleated sheet. Each layer composed of 3 to 5. Here the beta pleated sheet in yellow is containing 3 layers, and the other red is containing 4 layers. The two layers are held together by disulfide bridge. An adjacent strand, here is the adjacent strand connected to each other by short loop. Here is the loop. The heavy chain and light chain contain variable regions that are participating antigen binding site, and the C terminal region which is confer an immunity function. The heavy chain, the B region, is composed. Here is the variable region in heavy chain is composed of immunoglobulin, and the C region is composed of 3 immunoglobulin, 3 to 4 immunoglobulin. Each light chain is composed of one variable region, V region, not variable. Here is V region, immunoglobulin, and C region. Variable region are so named because their amino acid sequence vary among antibodies made by different B cell clones. The V region of one heavy chain, and the adjoining V region of one light chain form an antigen binding site. Here is the antigen binding site. Because the core structure unit of each antibody molecule contain two heavy chain and two light chain, then every antibody molecule has at least two antigen binding site. Here is the antigen binding site, and here is another antigen binding site. The heavy chain C region interact with other effector molecule and cells of the immune system. Here is the heavy chain region. The heavy chain exists in two forms. Here is the form and here is another form. One form, the heavy chain anchor membrane bound. Here is the membrane bound antibodies in the plasma membrane, and the other form is found only in secreted antibodies like IgG. Here is the secreted antibodies, which is contain no membrane bound antibodies. The C region of light chain don't participate in effector function and are not directly attached to cell membrane. Heavy chain and light chain are covalently linked by disulfide. Here is the disulfide bridge, and here is an experiment in rabbit. IgG is treated with enzyme bevine. Under condition of limited proteolysis, the enzyme acts on the Hing region. Here is the Hing region. Here is the Hing region, and cleaves the immunoglobulin into 3 separate pieces. Here is the 3 separate pieces. 2 of the pieces are identical to each other and consist of the complete light chain, variable light and constant light associated with variable heavy and constant heavy domain. This fragment retains the ability to bind antigen because each contains paired VL and VH domain, and they are called FAB. FAB is abbreviated for fragment antigen binding. The third piece here is composed of 2 identical disulfide linked peptides, each containing the heavy chain CH2 and CH3 domain. This base of IgG has the propensity to self-associate and to crystallize into lattice and is therefore called FC region. FC region is abbreviation for fragment crystallizable. This experiment provides the first evidence that the antigen recognition function and the effector function of immunoglobulin molecule are spatially separated. Then we want to talk about the structural feature of antibody variable regions. Most of the sequence difference and variability among different antibodies are confined to 3 short stretches in the V region of the heavy chain and the 3 stretches in the V region of light chain. These segments of greatest diversity are known as hypervariable regions. The hypervariable regions are each about 10 amino acid residues long, and they are held in place by the more conserved framework sequence that make up the immunoglobulin domain of the V region. The other name of hypervariable region is the complementary determining region, CDRS, proceeding from either the VL or the VH amino terminus. These regions are called CDR1, CDR2, CDR3, and all the CDR of both the VH segment and the VL segment are the most variable of the CDRs. Here is the domain, here is the VH or VL, and there are 3 stretches in the loop as we mentioned previously, and CDR1, CDR2, CDR3. Here is the diagram which represent the variability. The variability is very high in CDR3, then CDR1 and CDR2. The classes of antibody molecule are also called isotype, and are named IgA, IgD, IgE, G, and M. Here we want to say a note that this isotype is defined according to FC region. As we mentioned previously, the FC region is the F fragment crystallized And in human, IgA and IgG isotype can be further subdivided into closely related subclasses or subtype called IgA1, IgA2, Ig1, Ig2, Ig3, Ig4. In mice, there are another subdivision to IgA2, Ig2, to Ig2A and Ig2B. The heavy chain C region of all antibody molecules of one isotype or subtype have essentially the same amino acid sequence. And here is the other designed name, Greek alphabet corresponding to isotype. The IgA alpha 1 is called alpha 1, and IgD equal delta, IgE equal epsilon, and IgG equal gamma, and like that. In human, IgM and IgE, the C region contain 4 tandem immunoglobulin domain, and the C region of IgG and IgA and IgD contain only 3, not 4 like IgE and IgM. These domains are generally designated CH domain and are numbered sequentially from amino acid terminus to carboxyl terminus. Its name is CH1, CH2, and so on until CH4. The different isotype and subtype of antibodies perform different effector function, and that due to the different Fc receptor in different cell such as phagocytes, NK cell, and mast cell. An antibody isotype and subtype differ in their C region and there in what they bind to and what effector function they perform. And here is a figure that represents the binding of the antigen in CDRs, complementary determining regions. Here is the 3 stretches of VH and the 3 stretches region or loop of VL. Antibody molecules are flexible, permitting them to bind to different arrays of antigen like this here, if it is close to each other or apart from each other. And this flexibility is conferred in large part by Hing region. Here is the Hing region, located between CH1 and CH2 in certain isotype. The Hing region varies in length from 10 to more than 60 amino acid residues in different isotypes, permitting molecular motion between the CH1 and CH2 domain. There are 2 classes or isotype of light chain. The light chain called kappa and lambda that are distinguished by their carboxyl terminal constant region. In human, about 60% of antibody molecules have kappa light chain and about 40% have lambda light chain. And any change in this ratio between kappa and lambda chain in human, that means it must be B cell tumor, because the many neoplastic cells being derived from one B cell clone producing a single species of antibody molecule. The difference between the antibodies, the IgA 1 and 2, it is an dimer connected by J chain or joining chain. IgD is a monomer and it is not secreted, it is a membrane binded immunoglobulin. IgE and IgG is a monomer, and IgM is a pentamer, and here is secreted and membrane associated antibodies. They are differ in the amino acid sequence of carboxyl terminal end of the heavy chain C region. In the secreted form found in blood and other extra fluid, the carboxyl terminal portion is hydrophilic. This tail is hydrophilic. The membrane bound form of antibody contain carboxyl terminal stretches that include two segments, a hydrophobic alpha helical transmembrane region, the positively charged amino acid sequence bind to negatively charged phospholipid head group on the inner leaflet of the plasma membrane and help anchor the portion in the membrane. In membrane IgM and IgD molecules, the cytoplasmic portion of the heavy chain is short, only 3 amino acid residues in length. But in membrane immunoglobulin gamma and epsilon E molecule, it is somewhat longer, up to 30 amino acid residues in length. In the secreted secreted immunoglobulins molecule, all of it are monomeric. The multimeric immunoglobulin like IgM and IgA molecule are also contain an additional 50 kilodalton polypeptide called the joining chain, which is bind the multimeric molecule to each other. There are smaller difference sequence between antibodies from different individuals in the C region of immunoglobulin heavy and light chain, representing and reflecting that its inherited polymorphism in the gene encoding the C region. When a polymorphic variant found in some individual of a species can be recognized by an antibody, the variant are referred to as allotype, and the antibody that recognize an allotype determinant is called anti-allotypic antibody. And the difference between antibody V region are concentrated in the CDRs and constitute the IDU type of antibody. And antibody that recognize some aspects of CDRs of another antibody is therefore called anti-idiotypic antibody. There have been interesting theories that individuals produce anti-idiotypic antibodies against their own antibodies. Monoclonal antibody. A tumor of plasma cell, myeloma or plasmacytoma, like most tumor cellular origin, is monoclonal and therefore produce antibodies of single specificity. In most cases, the specificity of tumor-derived antibody is not known, so the myeloma antibody cannot be used to detect or bind to molecules of interest. However, the discovery of monoclonal antibodies produced by these tumors led to the idea that it may be possible to produce similar monoclonal antibodies of any desired specificity by immortalizing individual antibodies secreting cells from an animal immunized with a known antigen. The method relies on fusing B cells from an immunized individual, typically a mouse, with an immortal myeloma cell line and growing the cells under condition in which the unfused normal and tumor cell cannot survive. The resultant fused cell of B cell from an immunized animal that grow out are called hybridomas, the fused cell between B cell and tumor cell. Each hybridoma make only one immunoglobulin derived from one B cell from the immunized animal. The product of these individual clones are monoclonal antibody, each specific for a single epitope on the antigen used to immunize the animal and to identify the immortalized antibody secreting clones. And here is the mouse, which is injected by X antigen. Then this is a B cell from an immunized animal. It must be fused with myeloma cell in order to give the B cell the immortality. Then here is the hybridoma here. Each cell produces monoclonal antibody. And here is a common application of monoclonal antibody One of it is identification of phenotypic marker unique to particular cell type by defining a cluster of differentiation marker, then the immunodiagnosis and tumor identification. Also, for therapeutic purpose, some example includes antibodies against cytokine, tumor necrosis factor used to treat rheumatoid arthritis and other inflammatory disease, and antibodies against CD20 for the treatment of B-cell leukemias and for depleting B-cell in certain autoimmune disorder. Also, antibodies against epidermal growth factor receptor to target cancer cells, antibodies against vascular endothelial growth factor, a cytokine that promotes angiogenesis in patients with colon cancer, and so on. Other function is a functional analysis of cell surface and secreted molecules bind to cell surface molecule and either stimulate and or inhibit particular cellular function are invaluable tool for defining the function of surface molecules, including receptor for antigen. And here is some target for monoclonal antibody and its effect and the disease that must be treated. Synthesis, assembly and expression of immunoglobulin molecule. The maturation of B cell from bone marrow progenitor is accompanied by specific changes in immunoglobulin genes expression, resulting in the production of immunoglobulin molecule in different forms. The earliest cell in the B lymphocyte lineage that produce immunoglobulin polypeptide, called the pre-B cell, synthesize the membrane form of the mu heavy chain. This mu chain associated with protein called surrogate light chain to form the pre-B cell receptor, and a small proportion of the synthesis pre-B cell receptor is expressed on the cell surface. Immature and mature B cell produce kappa and lambda light chain, which associate with mu protein to form immunoglobulin mega molecule. Mature B cells express membrane form of IgM and IgD, the mu and delta heavy chain associated with kappa and lambda light chain. This membrane immunoglobulin receptor serves as cell surface receptor that recognize antigen and initiate the process of B cell activation. Here is the immunoglobulin expression during B cell lymphocyte maturation. The stem cell, none of the immunoglobulin has been expressed. Then pre-B cell, the pattern of immunoglobulin is cytoplasmic mu heavy chain and pre-B cell receptor here. Immature B cell immunoglobulin mega. Mature B cell have membrane with IgM and IgD. Activated B cell, low rate immunoglobulin secretion, but have chain isotype switching affinity maturation. Antibody secreting cells have high rate of immunoglobulin secretion and reduction of membrane immunoglobulin. Then the half-life of antibody. The half-life is the mean time before the number of antibody molecule is reduced by half. Immunoglobulin epsilon has a very short half-life of about 2 days in the circulation, although cell-bound IgE associated with the high affinity IgE receptor on mast cell have a very long life. See chapter 20. Circulating IgA has a half-life of about 3 days, and circulating IgM has a half-life of 4 days. In contrast, circulating IgG molecule have a half-life of about 21 to 28 days. The long half-life of IgG is attributed to its ability to bind the specific Fc receptor called the neonatal Fc receptor, FcRn, which is also involved in the transport of IgG from the maternal circulation across the placental barrier, as well as the transfer of maternal IgG across the intestine in neonate. Fc receptor N structurally resemble MHC type 1 or class 1 molecule described in chapter 6, and in the placenta and the neonatal intestine it transports IgG molecule across cell without targeting them to lysosome. In adult vertebrate, Fc receptor is found on the surface of endothelial cell, macrophage, and other cell type, and bind to micropinocytosed IgG in acidic endosomes. Fc receptor N doesn't target bound IgG to lysosome but recycle to the cell surface and release it at natural pH, returning the IgG to the circulation. This intercellular sequestration of IgG away from lysosome prevents it from being degraded as rapidly as most other serum proteins, including other antibody isotype, and as a result IgG has a relatively half-life. There are differences in the half-life of the 4 human immunoglobulin isotypes, but IgG1 and IgG3 are the most long-lived. Here is the Fc receptor neonate that prevents IgG. It binds to IgG in the pH 7, less than 7. In the blood, the pH is more it's about 7 pH. When the molecule of IgG is pinocytosed into acidic endosome, it must be selected by Fc receptor neonate and recycled to endosome, which is recycling endosome, and recycled to the circulation. Other component of acidic endosome must be degraded by lysosomes. Here is antibody binding of antigens, feature of biological antigens. Antigen is any substance that may be specifically bound by an antibody molecule or T cell receptor. Here we can see that antibody can recognize the antigen, whatever it is, metabolites like sugar, lipids, autocarides, and hormone, as well as macromolecule such as complex carbohydrate, phospholipid, nucleic acid. In contrast to T cell, which mainly recognize peptide. Although all antigens are recognized by specific lymphocyte or by antibodies, only some antigens are capable of activating lymphocytes. Molecules that stimulate immune response are called immunogen. That means immunogen is any molecule that can stimulate immune response. For example, the dinitrophenol compound doesn't make an immune response, doesn't stimulate immune response. That means dinitrophenol is not an immunogen. But if we conjugate dinitrophenol with carrier protein, it may become immunogen. Here, the dinitrophenol is called hapten, and the carrier protein called the carrier. The hapten carrier complex can induce the immune system. Here we have a note that antibody binds to only a portion of macromolecule, which is called a determinant or an epitope. The epitope and determinant is a synonymous, which is used interchangeably throughout this book. The macromolecule typically contain multiple determinant or epitope, some of which may be repeated and each of which by definition can be bound by an antibody. The presence of multiple identical determinants in an antigen is referred to polyvalency or multivalency. Most globular proteins don't contain multiple identical epitopes and are not polyvalent unless they are aggregate. The polyvalent antigens can induce clustering of B cell receptor and thus initiate the process of B cell activation. The special arrangement of different epitopes on a single protein molecule may influence the binding of antibodies in several ways. Here is we call it the overlapping, when the proximity of the antigen or epitope is near to each other. In rare cases, binding of one antibody may cause a conformational change in the structure of antigen, positively or negatively influencing the binding of second antibody at another site on the protein. By means other than steric hindrance, such interactions are called allosteric effects. And any available shape or surface on a molecule that may be recognized by an antibody constitute an antigenic determinant or epitope, Antigenic determinant may be delineated on any type of compound, including but not restricted to carbohydrate, protein, lipids, and nucleic acid. In the case of protein, the formation of some determinant depends only on the primary structure, and the formation of other determinant reflects tertiary structure, nor conformation. The epitope formed by several adjacent amino acid residues are called linear. Here is linear determinant. The linear determinant may be inaccessible in the native conformation and appear only when the protein is denatured. In contrast, conformational determinants are formed by amino acid residues that are not in a sequence, but become specially juxtaposed in the folded protein. Antibodies specific for certain linear determinant and antibodies specific for conformation determinant can be used to ascertain whether a protein is denatured or in its native conformation respectively. Proteins may be subjected to modifications such as glycosylation and phosphorylation, ubiquitination, acetylation, and proteolysis. This modification, by altering the structure of protein, can produce new epitopes. Such epitopes are called neoantigenic determinant, and they too may be recognized by specific antibodies. The structural and chemical basis of antigen binding. Structural and chemical basis of antigen binding. The antigen binding site of many antibodies are planar surface that can accommodate conformational epitopes of macromolecule, allowing the antibody to bind large molecule. Here is the molecule that involved in the binding is the 6 complementary determining regions, 3 from the heavy chain and 3 from the light chain. In a number of antibodies specific for small molecules such as monosaccharide and drugs, the antigen is bound in a cleft generated by the close apposition of complementary determining region from the variable light and variable domains. The recognition of antigen by antibody involves non-covalent reversible binding. Various types of non-covalent interaction may contribute to antibody binding of antigen, including electrostatic factor, hydrogen bond, van der Waals force, and hydrophobic interaction, depending on the structure of the binding site of the individual antibody and of the antigenic determinant. Also, the strength of the binding between a single combining site of an antibody and epitope of an antigen is called the affinity of antibody, and the affinity is commonly represented by this association constant Kd, which indicate how easy it is to separate an antigen antibody complex into its constituent. The smaller Kd indicate a stronger or higher affinity interaction because a lower concentration of antigen and of antibody is required for complex formation. The polyvalent antigen will have more than one copy of a particular determinant. Although the affinity of any one antigen binding site will be the same for each epitope of a polyvalent antigen, the strength of attachment of the antibody to the antigen must take into account binding of all the sites to all the available epitopes. This overall strength of attachment is called the avidity, and is much greater than the affinity of any one antigen binding site. Thus, a low affinity immunoglobulin mega molecules can still bind tightly to a polyvalent antigen because many low affinity interaction up to 10 per immunoglobulin mega molecule can produce a high avidity interaction. Here is the valency of interaction between one antigen binding site to the antigen is called affinity, and here is the avidity. Here the avidity is represented here in IgM, and the affinity is represented here. Precisely, the affinity is the attachment of one antigen binding site to one epitope. But the avidity is the collection of any antigen binding site that belonging to one molecule. If a polyvalent antigen is mixed with a specific antibody in a tube, the two interact to form immunocomplex. At the correct concentration, called a zone of equivalence, antibody and antigen form an extensively cross-linked network of attached molecules, such that most or all of the antigen and antibody molecules are complexed into large masses. Immunocomplex may be disassociated into smaller aggregate, either by increasing the concentration of antigen. Here is the increasing the concentration of antigen, so that free antigen molecule will displace antigen bound to the antibody, zone of antigen excess, or by increasing antibody molecule will displace bound antibody from antigen determinant, which is zone of antibody excess. If a zone of equivalence is reached in vivo, large immunocomplex can form in the circulation. Immunocomplex that are trapped or formed in tissue can initiate an inflammatory reaction, resulting in immunocomplex disease. Structural functional relationship in antibody molecules. The features related to antigen recognition: firstly, the specificity. The antibodies can distinguish between small and difference in chemical structure. Some antibodies produced against one antigen may bind to different but structurally related antigen. This is referred to as a cross-reaction. Antibodies that are produced in response to microbial antigen sometimes cross-react with self-antigen, and this may be the basis of certain immunologic disease. Then, secondly, the diversity. The ability of antibody in any individual to specifically bind a large number of different antigens is a reflection of antibody diversity. And the total collection number of antibodies with different specificities represent the antibody repertoire. The genetic mechanism that generates such a large antibody repertoire occur exclusively in lymphocytes. This diversity is generated by random recombination of a limited set of inherited germline DNA sequence to form functional genes that encode the variable region of heavy and light chain, as well as by the addition of nucleotide sequence during the recombination process. The millions of resulting variation in structure are concentrated in the antigen binding hypervariable region of both heavy and light chain, and thereby determine specificity for antigen. Thirdly, the affinity maturation, a mechanism for the generation of high affinity antibody involves subtle change in the structure of the variable region of antibody during T-cell dependent humoral immune response to protein antigen. These changes can come about by a process of somatic mutation in antigen-stimulated B lymphocytes that generate new variable domain structure, some of which bind the antigen with greater affinity than did the original variable domain. Those B cells producing higher affinity antibody preferentially bind to antigen, and as a result of selection becomes the dominant B cell with each subsequent exposure to the antigen. This process, called affinity maturation, results in an increase in the average binding affinity of antibody for an antigen as a humoral immune response evolves. Thus, An antibody produced during a primary immune response to a protein antigen often has a key day in the range of 10 to 7 to 10 to 9 molari. In secondary response, the affinity increases with the key day of 10 to 11 molari or even less. Feature related to effector function. Many of the effector function of immunoglobulin are mediated by the Fc portion of the molecule. An antibody isotype that differ in this Fc region perform distinct functions. Immunoglobulin goes microbe and target them for phagocytosis by neutrophil and macrophage. Immunoglobulin epsilon bind to mast cell and trigger their degranulation because mast cell express IgA specific Fc receptors. Another Fc-dependent effector mechanism for humoral immunity is activation of the classical pathway of the complement system, initiated by the binding of a complement protein called C1q to the Fc portion of the antigen complex IgG or IgM. Here is the change in antibody structure during humoral immune response. It can be increase of affinity due to the increase of structure here, from here to here. But if they are changed from membrane to secreted, there are no change in structure. Also, if they are isotype switching, there are no change in the affinity. So this is the last in this chapter, and goodbye.